Good afternoon, everyone. My name is Laura Lambert, and I'm the director of the Functional Omics Resource at Mayo Clinic. I would like to welcome you to the Rare Disease Speaker Series. This is a joint venture between the Center for Individualized Medicine at Mayo Clinic and the Mellowes Center at MCW. This lecture series is an opportunity to highlight the work being done around rare disease. It has been my pleasure to introduce today's presenter, Dr. Mark Osborn. Dr. Osborn is an associate professor at the University of Minnesota Medical School in the Department of Pediatrics in the Division of Blood and Marrow Transplant and Cellular and Gene Therapy. His lab investigates and applies genome editing technologies for precision sequence modification for rare diseases. This work employs approaches for individualized medicine, as well as targeted genome strategies for large genetic cargo insertion that is broadly applicable to rare disease patients. Today's presentation is titled Genome Editing for Rare Disease. Throughout this talk, please use the Q&A in Zoom to submit your questions. We have planned time for Q&A at the end of the session. Now please join me in welcoming Dr. Mark Osborn.
Thank you so much. Thank you so much for the kind introduction, Dr. Lambert, and for the opportunity to be here. Good afternoon, everyone. Again, thank you for the opportunity to be here today. I'm Mark Osborn from the University of Minnesota. And I always enjoy talking about one of my passions, rare diseases, that I put in quotation marks because at the University of Minnesota in MCW and at Mayo, rare diseases are not so rare because people come here to be treated. And so it's an opportunity to interact with people who have conditions that are often at the vanguard for advancing new gene and cell therapies. And so it's exciting to be able to talk about this today because while rare diseases are often monogenic, they're incredibly complicated because often they have a cascade of events driven by a pathogenic nucleotide variant. And so my laboratory's goal is to harmonize all of these equally complex therapeutic options, such as large data sequencing, et cetera, and use that to inform best practices for treating rare diseases. And the focal point of my laboratory is using gene editing to do that both for personalized medicine as well as then advance that from an N=1 approach to more broadly for N=some and excuse me, hopefully for N=all, where we can create a suite of genome editing platforms that can be broadly applied for therapeutic and curative intent. So throughout my career, I've had the opportunity to work with the major gene editing platforms, meganucleases, zinc finger nucleases, TALENs, and CRISPR-Cas9. I show this just because all are still relevant, and they all have the same commonality of being programmable for a specific nucleotide sequence that then co-localizes and then modifies the DNA in some fashion. So the condition I'm going to spend the majority of my time talking about today is a rare disease called recessive dystrophic epidermolysis bullosa. And because Dan Voitos, the co-discoverer of the TALEN technology, resides at the University of Minnesota and as a good friend, enabled us to custom design TALENs for a gene target that is collagen 7. Dystrophic epidermolysis bullosa, or EB, is caused by loss of function mutations to the collagen 7 gene. Collagen 7 participates in assembling and anchoring the outer layer of the skin to the underlying dermis. And so it's a crucial functional protein that its loss essentially becomes very similar to a burn. And so single nucleotide changes, insertions, deletions, all loss of function mutations can result in this severe phenotype due to loss of collagen 7 functionality and its inability to then anchor the outerlying architecture of the skin to the underlying dermis. So with Dan Voitos and his labs group, we had the opportunity to custom design, so individualized medicine approach to create TALENs for a patient seen at our institution who was treated by bone marrow transplantation. And the goal of that therapeutic strategy was to repopulate the bone marrow compartment particularly with mesenchymal stromal cells that are a source of collagen 7. They also have migratory properties and orchestrate wound repair. And so because allogeneic therapies are associated with transplant-associated risks, such as graft versus host disease, we assessed whether we could employ TALENs for individualized genetic medicine to correct this patient's collagen 7 pathogenic nucleotide variant. His particular DNA change was an installation of a premature termination codon in the collagen 7 gene that resulted in a severe phenotype. And so we generated TALENs and provided a correction template donor in trans in fibroblasts obtained from a skin biopsy from this patient, and then analyzed them and showed that we could incorporate the donor and correct the pathogenic variant that then resulted in restoration of collagen 7. This patient was a true null. So at the bottom, the native state is that when interrogated with collagen 7 antibodies, these patients are non-reactive. And then post-TALEN treatment, both at the molecular level and at the protein level, we observed restoration of the reading frame with production of the collagen 7 protein. This particular phenotype, because it involves the skin, our approach was intentional in obtaining fibroblasts, certainly for their ease of obtainment, but they're also therapeutically relevant as an autologous source of gene-corrected cells. And so there are clinical trials where fibroblasts have been expanded and applied topically for patients. We also proceeded through a route of reprogramming those fibroblasts to pluripotency. As I mentioned, mesenchymal stromal cells are a potential therapeutic source of collagen 7. And so we corrected the fibroblasts and then reprogrammed them to pluripotency. And one of the things we've observed and act as a good three-dimensional skin surrogate was that teratomas will form skin-like structures. And so we had injected the gene-corrected or uncorrected pluripotent stem cells into immune-deficient animals and then harvested them and looked for skin-like structures because the teratomas form all three layers of the germ layer, including ultrastructural components of the skin. And so then when we stained the skin for cytokeratin, which is an epidermal protein, and then the collagen 7, we showed deposition of that at the correct position, which is the dermal epidermal junction. So this represented a functional cellular cure. At this time, TALENs were the state of the art, and they operate through binding proximal to the target location, and then introducing a double-stranded DNA break. For this particular rare disease and some of the other ones we study, they also have a predisposition for tumors. So the severe phenotype in EP results in constant wound and healing, which causes proliferation, inflammation, and often the development of very aggressive squamous cell carcinomas. And so using nuclease technology allowed us to achieve our goal, but it also comes at the expense of introducing double-stranded DNA breaks. And while we tend to look at those as binary, where they proceed through an error-free pathway, which is the pathway we took by delivering a repair template in trans, what it also can proceed is through error-prone, where it resects the double-stranded DNA breaks and then ligates the free ends back together, and that is often associated with insertions and deletions. And so while we tend to look at this as an orderly process where it's an either or, it's actually a much more complicated process where it's both events occur, meaning that a double-stranded DNA break often has a mixture of homologous recombination, which is the desired event for gene correction, but there are also competing error-prone, and so this becomes a mixed population. And again, given the phenotype where they have a magnified risk for tumorigenesis, maintaining the integrity of the DNA is an important consideration. And so as the capability to program reagents for the genome advanced new technologies beyond TALEN nucleases and CRISPR-Cas9 nucleases emerged. And these are the base editor and prime editor platforms that allow for sequence-specific modification, but they don't introduce the significant amount of double-stranded DNA breaks. And so it becomes a powerful platform for genetic medicine. And so, excuse me, we had the opportunity for this patient, EB patient, as well as another with premature termination codons to ask the question, could we apply cytosine base editors or adenine base editors? So cytosine base editors directly convert cytosine to thymine through a deamination event. Likewise, adenine base editors remove the adenine amine group, and that's viewed as an inosine or guanine by the cell. So it allows for C to T or A to G transition mutations. And again, using the platform for epidermolysis bullosa, what we did was targeted the opposite strand. So there's a premature termination codon, and by targeting the opposite strand, we were able to apply and ask the question, can an adenine base editor revert the sequence back to wild type? And so shown here is the experimental design using an adenine base editor. An important advance as well is that it no longer requires a repair template because the editor itself modifies the DNA directly. And going down a similar path, we used fibroblasts from the patient I shared previously, as well as another patient. Ask the question, could we introduce adenine base editors to the fibroblasts, differentiate them into pluripotent stem cells as well as mesenchymal stromal cells, and show restoration of collagen 7? So again, this is a true null patient. Shown is a photomicrograph of a skin biopsy from a wild type and EB patient. At left is the red line showing the deposition of collagen 7 at the dermal epidermal junction, and then the EB patient is a true null for this. We had an early version of the adenine base editor. The constituent is Cas9 directs programmable localization of the complex. And then there's an adenine deamination domain that mediates the adenine to guanine conversion by deaminating adenine. So we electroporated this into the primary cells as a messenger RNA molecule, and then use Illumina sequencing to assess correction frequency at the genomic and messenger RNA levels. We saw roughly 22 to 25% reversion of the genomic base from the premature termination codon back to the wild type base. And then in the messenger RNA, we saw nearly double amount of the edited population. We think that because this is a patient with a premature termination codon, likely subject to nonsense-mediated decay, is that when we correct the base, it results in a more stable transcript. And that's why there's the disparity between what we see at the genomic level as well as the messenger RNA level. Upon immune fluorescence microscopy of the treated fibroblasts, and these are fibroblasts, which I should note, for the TALENs, what we did was we adopted a enrichment strategy using drug selection. So our donor also had a pyromycin drug cassette in it that would allow us to obtain gene-corrected cells. An important operative aspect of the base editor platform is that it doesn't require a donor and often operates at high enough frequency where enrichment isn't required. So these data show a bulk population that is electroporated and does not undergo selection, and shown at the top is the untreated patient. In the middle panel then below shows adenine base editor resulted in correction of the base and restoration of collagen 7 expression. Similar to the TALEN work, what we did then was induce these to pluripotency and inject them into animals and ask the question, do they form collagen 7 in vivo in the three-dimensional skin architecture? Similar to TALENs, they did do that and shows ultrastructural deposition and reconstitution in vivo. We then asked the question, with pluripotent stem cells, could we differentiate them into mesenchymal stromal cells, which are being applied clinically for EB, gene-corrected cells reprogrammed, and then formed mesenchymal stromal cell populations in vitro, similar to those from wild type bone marrow. And so they retain their ability to form, and then also the mesenchymal stromal cells as a source of collagen 7, express collagen 7 by analysis by immune fluorescence. So these showed that we could, in the absence of a donor, correct the base. And importantly, when we analyzed insertions and deletions, and so evidence of breakage of DNA. So the contrast between the TALEN and the base editor is that the base editor is based off nicking the DNA strand, a single strand, rather than introducing double-stranded DNA breaks. And so it would be anticipated that there's a fewer accumulation of mutagenic insertions and deletions. And that was the case we observed in our primary cell population, that the insertions and deletions and evidence for double-stranded DNA breaks was below 2%. I mentioned that our platform for this was to deliver messenger RNA by electroporation. And that results in a fairly extended expression profile, particularly in fibroblasts, which are slow growing, so that we see evidence for expression of messenger RNA introduced by electroporation well beyond 2 weeks. And so what we did then, because the reagent is programmable for the collagen 7, doesn't necessarily mean that it won't have off-target effects, meaning that it binds elsewhere and can act upon that sequence based on some homology to where CRISPR-Cas9 binds. And so we found an off-target site where we did see evidence of adenine deamination. And we think that is due to some promiscuity between where CRISPR-Cas9 binds itself. What we also observed was some on-target bystander editing. And so the window for base editing is not necessarily single nucleotide resolution. It happens within a window within the guide RNA. And that's what we observed, is that other adenines within the targeting window and proximal to the mutation target base were also modified, but to a lower degree than the target base. And so this highlights that the extended expression profile also promotes these potential on and off target bystander edits. Toward resolving this, we sought to merge this with technologies, further technologies coming out of David Liu's laboratory at MIT, which is to package the base editor as a peptide rather than a messenger RNA and deliver it to cells. And the technology they advanced was essentially replacing the genetic cargo of a lentivirus with the protein of a base editor. And so all of the genetic elements are stripped out of the virus, and it becomes a protein shell that carries the base editor and can deliver it based on the transduction capabilities of the virus, which is pseudotyped with a VSVG envelope. And so it becomes a protein delivery vehicle rather than an mRNA electroporation strategy. So these are the data I showed, that with electroporation messenger RNA, we saw roughly 25% editing in the genomic DNA, twice that in the messenger RNA, but with a corresponding on-target bystander editing within the collagen 7 locus, as well as off-target editing. However, when we transition to the use of virus-like particles for the same site and passively perform gene transfer, meaning that we pipetted these onto monolayer cells, and the transduction capabilities resulted in cellular entry, uncoding, and delivery of the base editor, is that we could now dramatically improve our editing frequencies to nearly saturating levels. And because it becomes a hit and run vehicle where the protein itself has a finite lifespan that is non-renewable and is relatively quickly degraded, is at the off-target site, off-target events, both for adjacent base editing at the target site as well as true genomic off-target sites, was essentially diminished back to background. And so this becomes a transient hit and run delivery vehicle that offers greater activity as well as greater safety. And so we've applied A base editors for collagen 7. Others in the field have used cytosine base editors. Both technologies are available for use with virus-like particles. It offers greater specificity, greater activity without requiring a donor molecule. And so these synergize to become efficient delivery platforms that enable target site-specific locus modification using these editor platforms. So shown at bottom left is the mutational constellation of collagen 7, where many patients often have a disease pathogenic variant that's unique to them. And only a subset are transitions, so amenable to C to T or A to G transitions and the use of the base editor, where many others are insertions, deletions, or transitions, which are not amenable to the architecture of the base editor. And so looking at the graph at bottom is a pie chart of the composition of collagen 7 driver mutations and which editor platform might be applicable. And so the prime editor is applicable to nearly all of the reported mutations. And while the prime editor is an architectural advance from the base editor, they're both centered around the use of Cas9 to confer programmability, while instead of a deamination domain, the prime editor now has a engineered reverse transcriptase that is fused to Cas9. And so then using a engineered prime editor guide RNA or EPEG RNA that has a scaffold that allows Cas9 interaction, a protospacer that directs DNA binding. It also has a RT template, a reverse transcriptase template that contains the user-defined edit, and then the primer binding site where the reverse transcriptase binds and starts to perform its duties as a DNA-dependent RNA polymerase and writes in the edit that is then installed into the genome. A corresponding downstream nick then results in faithful installation of that. And so the Cas9 unwinds the DNA through the programmability and the interaction of the scaffold, which allows Cas9 to find its target. The EPEG RNA then writes in off of this RNA template a specified edit that occurs from priming off the primer binding site, and then a downstream guide RNA nicks the complementary stand and essentially seals in and locks in the edit and allows for all of the local DNA modifications, transitions, transversions, insertions, and deletions. And so this is nice work from molecular therapy about 2 years ago, where a group employed the prime editor for a non-targetable collagen 7 mutation through other means, such as the base editor. And so they developed prime editor reagents, introduced them into normal human dermal fibroblasts. Patient fibroblasts shown at left is the normal human dermal fibroblasts, and then the patient cells in the middle are untreated, which similar to ours are a true null for collagen 7, and then post prime editing to restore the mutated base, they saw restoration of collagen 7. So across the spectrum of personalized medicine, the field has been able to use TALENs base editors and prime editors based on the architecture of the platform. Importantly, for base editors and prime editors, that does not require donor molecules as an important safety advance, because these employ the nickase version of Cas9. They don't introduce high rates of double-stranded DNA breaks as opposed to TALEN nucleases or CRISPR-Cas9 nucleases. The challenge becomes, from a personalized medicine perspective, particularly for collagen 7, is reagent development at the individual level, particularly for the prime editor platform, where the prime editor guide RNAs can be challenging to design, develop, and obtain a high frequency editor. And so with the ability of the prime editor to write larger sequence modifications, we sought to ask the question, could we use it for broader applicability? So moving now from an n=1 to an n=some concept. And this was based on the unique architecture of the genes such as collagen 7, genes such as dystrophin that accommodate or tolerate deletions of exons, because exons can be removed and the upstream exon then fuses with the downstream exon in frame to create an in-frame deletion. And these can have partial functionality. And so collagen 7, the majority of the exons could be deleted to create an in-frame fusion. And so we asked the question, could we delete pathogenic exons rather than necessarily directly correcting a patient-specified base? This has been done previously with CRISPR-Cas9. This is data showing 2 guide RNAs on opposite sides of exon 80 that contained an insertion mutation. Electroporation of the ribonucleoprotein complexes resulted in excision of exon 80 and restoration of collagen 7 expression. Similar to the previous data is that this occurred with a stochastic repair of the double-stranded DNA break. So a mixed populations of insertions and deletions were introduced, and these are uncontrollable, is that this profile is different and becomes concerning, again, particularly for the introduction of double-stranded DNA breaks in any condition, particularly those that have a magnified risk for tumorigenesis. So the prime editor is a unique architecture in that 2 prime editor guide RNAs, so conceptually similar to the previous example where they can flank a target site, but rather than introducing double-stranded DNA breaks, the prime editor guide RNAs are engineered such that they overlap with one another, and then they essentially write over the target site and remove it. And so this is accomplished initially targeting the dystrophin exon 51, which is able to be removed and restore dystrophin, and showed that a number of prime editor guide RNA candidates could be used to accomplish this deletion. And importantly, this deletion happens without double-stranded DNA breaks or insertions and deletions. And so it's a seamless removal of a target site. And we adopted this procedure to ask the question, could we do this in primary cells? The previous data was in cell lines. So targeting the mutation exon rather than the mutation itself was our strategy by flanking the entire exon for removal in primary fibroblasts using 2 prime editor guide RNAs, and then going through our experimental design where we did this in fibroblasts and then induced pluripotent stem cells differentiated into therapeutic cell populations. Keeping pace with the advances in the field. So the original prime editor used a wild type version of the reverse transcriptase. David Liu's group, with an eye toward better deliverability of this system, has streamlined and increased the activity of these molecules by using directed evolution and smaller reverse transcriptase enzymes to achieve a suite of reagents that can be employed for better activity with smaller size. And so we did a comparison of the prime editor PE Max, which is the first version of the enzyme, and then the latest PE6 versions, and did a head-to-head comparison in our collagen 7 exon targeting strategy. So the reagents, again, flank the target exon. The prime editor guide RNAs overlap with one another to essentially write over that exon and remove it without the introduction of double-stranded DNA breaks. We tested the prime editor Max and then the different PE6 suite of enzymes, and similar to the original publication, observed that PE6C and PE6D, which have different architectures and sizes for the prime editor itself, mediated high levels of deletion in cell lines. We then assessed the deliverability using an all plasmid approach or an all RNA approach, and showed that when the prime editor is delivered as messenger RNA and the prime editor guide RNAs are synthetic RNA transcripts are delivered to primary cells, showed the highest levels of deletion shown in the agarose gel. So the bottom band is the deletion product, showing that when possible, using all RNA delivery mediated the highest level of exon deletion. As I mentioned, that this exon we targeted is dispensable for its loss is dispensable, meaning that when it's removed, the upstream exon becomes in frame with the downstream and can produce a peptide. And so that's what we observed in that unedited cells. Again, this is a true null patient. When we deleted the target exon, we then saw restoration of collagen 7 by immune fluorescence as well as Western blot. Notably, the deletion is fairly small and the peptide is fairly large, so there's not a overt size difference available on Western blotting, but we think that that's the size difference between the relatively small exon deletion and the large size of the collagen 7 peptide itself. We saw robust editing frequencies in induced pluripotent stem cells. So rather than reprogramming the fibroblasts to pluripotency, we directly modified the genome of iPS cells from this patient. And at over 90% editing frequency in colonies that were derived from single cell clones, we showed editing, which is the deletion product shown in the green arrows at bottom, and then use those as, again, the platform to derive mesenchymal stromal cells and, in this case, keratinocytes. So we were able to accomplish this workflow, and again, importantly, moving from an individual patient medicine approach to now a more regional one, where the literature suggests rather than the individual patient who had a exon mutation, there were 18 patients that have mutation in this exon. So it goes from being a single-use reagent now to more broadly applicable. The architecture, though, of collagen 7 and dystrophin are unique in that aspect, in that there are exons that can be deleted with restoration or maintenance of the open reading frame. And so it does require a specialized genomic accommodation and architecture. And the prime editor guide RNAs can be burdensome to detect. This is a question I often get. And for this particular project, it was essentially the second disease candidate we pursued. Another disease candidate, we built over 150 prime editor guide RNAs with 0 success in showing installation of a prime edit. We then asked what the efficiency for the collagen 7 was, and it was well over 75% of the engineered prime editor guide RNAs showed functionality. And so there's a great discrepancy for individual use and development of prime editor guide RNAs. And so seeking to uniform a uniform process where we can use a suite of well-characterized, highly active prime editor guide RNAs to modify the genome, to create a landing pad where then a large cargo could be inserted. So now from personalized medicine to more broadly applicable and loss of function mutations could benefit from a single treatment by recapitulating the entire open reading frame. That's the entire basis for lentiviral gene transfer. Again, the off-target considerations for that become that a gene delivered by a lenti, AAV, or adeno doesn't necessarily have the locus-specific regulatory elements, and so there's typically supernormal protein expression, which could potentially be deleterious. There's both immune potential side effects from the viral vector itself, as well as viral vectors that integrate into the genome, can cause collateral genomic and transcriptomic aberrations, resulting in oncogenic integration. And the standard gene therapy for epidermolysis bullosa is a viral vector system derived from herpes simplex virus called BVEC that delivers 2 copies of the collagen 7 gene under non-native regulatory elements that's applied topically. It does not integrate into the genome, making it low oncogenic risk, but because it is not a permanent genetic event, it also may require redosing, and that could be problematic for redosing, both from an engineering, potentially immune, as well as a patient health benefit. So those risks associated with viral vector transgenesis to incorporate full-length genes. Again, I've talked about the potential side effects from introducing double-stranded DNA breaks, balance versus the advances with A-base editors and prime editors that diminish those double-stranded DNA breaks, but can accommodate large sequence modifications that are prevalent in collagen 7. And so one strategy to maximize the benefit of prime editors to accommodate larger sequences on the order of 30 to 50 nucleotides is to use that to introduce attachment sites in the genome as a landing pad for a subsequent larger cargo insertion. So these attachment sites are roughly 38 base pairs. They're derived from phage recombinases, and can mediate a unidirectional recombination event between paired at P or at B sites. And so the principle is to use prime editing to insert attachment sites in the genome and then provide a donor with a large cargo to undergo unidirectional and programmable insertion into the genome. So the first efforts for this used a Cas9 reverse transcriptase BXB1 integrase or recombinase. That's the enzyme that mediates the unidirectional modification or recombination between ATB and ATP for targeted insertion of large cargo into the genome. The Liu laboratory subsequently made advances using their prime editor system and 2 prime editor guide RNA, similar to the concept that I showed, is that while they can be overlapping and replace sequences, they can also be overlapping and introduce sequences. And so they can be engineered to insert these attachment sites into the genome, and then a recombinase and a donor molecule are provided in trans, and that entire system then allows for programmable insertion of this large cargo into the genome. So the prime editor is used to install and create this landing pad in the genome by introducing these attachment sites, followed by the recombinase and a donor molecule with a paired attachment sequence for seamless double-strand break-free insertion into the genome. So building off of our exon deletion work, one of the questions we asked is, could we replace that exon? Our prime editor guide RNA was such that exon deletion occurred simultaneous with ATB insertion. So in another set of experiments, either sequentially, where the AT site was introduced, followed by a second gene transfer event, or a one-pot strategy where all of the materials, the prime editor guide RNA, prime editor enzyme, the recombinase, and donor molecule were all delivered in a single electroporation event, were pursued for reinserting our target exon in collagen 7, exon 5, back into the genome. And we were able to show recombination. So the middle panel of Sanger sequencing, the blue site is the attachment site, and then the post-recombination product converts the genomic at site to a new called an at L or at left site, which has a distinct sequence signature. And so we were able to demonstrate recombination, and then to better track insertion of our donor sequence, we introduced a number of silent mutations so that we could assess it. And so the individual black arrows at bottom are codon optimized collagen 7 sequences that have now been reinstalled back into the genome to reconstitute exon 5. What we observed with this was that efficiencies were low, particularly in primary cells. While we could detect the event at the molecular level, the occurrence was relatively low. And so that prompted work by the Liu laboratory to advance the system similar to what they had done previously to use directed evolution to create new Cas9 and reverse transcriptase molecules. Now they unleash the power of their in vitro evolutionary campaign led by David Liu's group, Smriti and Daniel and his group, to modify the recombinase. So the recombinase appears to be the rate limiting step in the installation of these sequences. And so they instituted an evolutionary campaign for the BXB1 recombinase. They identified a number of fitness mutations that increased activity and prioritized 3 that are shown in the red boxes to create what now is an evolved and engineered or EEBXB1 that we then assess for the prime editor site-specific integrase genome editing or passage. So comparing the recombination elements versus the different iterations, both of technologies and previous versions of the BXB1 showed an increase in integration efficiency using the evolved enzyme and then the EE or evolved engineered enzyme. Shown at left is passage, which is the first generation of this particular procedure. Shown at right is paste, which is the early version using the Cas9 reverse transcriptase BXB1 fusion construct. And so this new evolutionary campaign resulted in increase in genomic integration efficiency at 2 loci in the genome, and importantly, could accommodate a broad range of genetic cargo. Up to 10.5 kb was tested. Of note, collagen 7 as a large cDNA construct is roughly 9 kb. And so this was very, very exciting to us because it was well within the capability of this system to potentially introduce a gene the size of collagen 7. And these data are in cell lines. And so this was one potential solution to the hurdle, is that the BXB1 enzyme is rate limiting. And so to start to optimize the procedure is that in cell lines, all of the material could be delivered as DNA. In primary cells, that was prohibitive and associated with significant toxicity. And so what we worked with were alternative donor DNA formulation delivery templates. And so the experimental design was to deliver the prime editor and recombinase sequences by electroporation. And the prime editor installs the genomic landing pad. The recombinase then can accommodate insertion. And so then what we did was, between 2 and 4 hours later, was introduced a donor with an integrase deficient lentiviral particle. And so it became a means to use the properties of the virus to deliver the repair template. And this increased efficiencies in primary cells at 2 loci that we have interest in, collagen 7 primarily, and increased our targeting efficiencies to about 30%, which prior to that we think was below 1%. So this was an exciting advance that now positions us to install these sequences into the genome. And so that's the state of where we're at, is we've made these architectural and delivery advances that we're pursuing for large cargo insertion. I also want to spend a minute, because all that work I presented was in vitro and ex vivo, and I also want to talk a little bit about in vivo targeting for another rare disease, lysosomal storage disease, mucopolysaccharidosis type 1, that again has a relatively large mutational spectrum, but the principles of being able to create these genomic landing pads becomes exciting for this because it enables the installation of a genetic element and again, much appreciation to Dr. Lambert, who created this mouse, which has a targetable mutation in the genome, premature stop codon, that we've applied adenine base editors. And one thing I want to highlight here is that because of the size of the editor, we had to split it across 2 adeno-associated viral vectors that were then injected into postnatal day 1 pups. We saw significant editing in the liver for this particular single nucleotide polymorphism. What we're interested in, and then that resulted in protein expression and decrease of lysosomal storage disease burden. And what we're excited to do with this is now merge the concept of being able to deliver these components through AAV gene transfer with the concept of large cargo insertion. There's a clinical trial underway using zinc finger nucleases to generate a double-stranded DNA break proximal to the albumin promoter to insert the full-length transgene for mucopolysaccharidosis type 1 and 2. The principle behind that is to do this in vivo, and albumin is a highly expressed gene in the liver. And so by inserting a therapeutic cargo into the liver, it essentially can employ the local regulatory elements to create high levels of this enzyme. And this is an enzyme where supernormal levels are well tolerated and are associated with increased therapeutic benefit. And so the principle becomes transitioning from being able to perform the DNA break-free large sequence installation ex vivo and use the concepts being explored with nucleases in vivo to synergize those 2 components to avoid double-stranded DNA breaks, creating a safer therapy that could be delivered in vivo for liver-targeted integration of, in this case, the lysosomal storage disease transgenes. So it becomes an exciting platform as a more targetable and broadly applicable therapy. So in conclusion, we've had the opportunity to partner with some amazing people to apply the adenine, cytosine, and prime editors, both for individual patient mutation correction, as well as then more broadly moving from individualized medicine from n equals 1 to n equals some to n equals all. The architectural advancements mean that donors are often not required. When we do these large cargo insertions, we do have to provide a donor, but with the ability to modify and use differential templates such as integrase deficient lentiviral vectors, we can deliver the cargo in a more efficient manner to get double-stranded DNA break-free insertion. And so the prime editor really offers broader capabilities for patient-specific targeting, transitions, transversions, insertions, and deletions, but also for creating these genomic landing pads as a broadly applicable strategy that we're pursuing for locus-specific integration of cDNAs upstream of every reported downstream mutation. So it becomes a universal reagent that we hope will be broadly applicable. So I've had the amazing opportunity to work with the Liu Laboratory at MIT, my own group. I'm grateful for colleagues at MCW and Mayo have just been fantastic partners too. And so I'm grateful for the team effort and the opportunity to share these data with you today. And I'm happy to take any questions, and I appreciate everyone's attendance today. So thank you so much.
Thank you for that amazing talk, Mark. That was phenomenal. I was so glad to have the opportunity to learn more about your work. I'd like to lead off the Q&A. We have a couple in the chat, but I wanted to ask you, what your thoughts are on the landscape of getting these base editors and prime editors approved for therapeutic use. I know we're working as a field toward getting a platform approval in place for CRISPR. Do you think this would fall under that, or it's going to need its separate regulatory element?
I think that's a great and exciting question, and I'll credit Fyodor Urnov for the n=1 to n=some to n=all. That's a phrase he coined, where I think that's the goal is, can we achieve essentially a CRISPR regulatory approval platform, and then the A base editor, prime editor, et cetera, are not considered new investigational drugs. And I know that's a huge effort of his, and I think that's an exciting prospect that I hope that the field will allow, rather than each locus or even each individual platform to build regulatory approval around CRISPR-Cas9. I think would streamline the path to clinic. I think it represents an exciting strategy to accomplish that.
Yeah, I completely agree. I'm looking forward to the next few years, seeing how far we can get. So I'll move to the questions from the chat now, starting with, how common are gene alterations in the promoter or intronic regions of COL7A1 in RDEB patient cohorts?
That's a really good question. I apologize, I don't have the exact number, other than to say we have not seen promoter alterations. We commonly see splicing variations, and so intronic mutations are common. So what the proportion is to open reading frame, I apologize, I don't have that data, but they are a common occurrence. And so targeting introns as well as exons is part of the requirements for addressing collagen 7.
Great. Yeah, that kind of touches on part 2 of that same question. What strategies should be considered for gene editing in these non-coding regions to ensure effective correction of the gene?
Yeah, I think that is almost architectural, meaning that I think there's been some exciting work using base editors to disrupt splicing. And so it might be, if it were at intron-exon boundary, could potentially be targetable for skipping an exon. So the data I shared, where we know some exons are dispensable, that could be a potential strategy. I think that prime editing to restore a splice site becomes really guided by the local architecture and constituency of the gene product. So a lot of the base editor work we do still employs the canonical NGG PAM requirements of CRISPR. There's certainly a number of different PAM variants. What's exciting about the prime editor is it opens up the targetability window, is in that it, similar to previous nuclease approaches, where the goal was to introduce a double-stranded break proximal, say within 50 to 100 nucleotides of the target base. I think the similar concept for the prime editor can be accommodated, so you could keep the NGG requirements, but position it at a farther site from the mutation itself, but within a window that allows the prime editor guide RNA to rewrite or overwrite that potential target base. And so I think that the flexibility either of PAM variants or the prime editor really, and in the context of what the local sequence composition is, really guides the design components.
Great. Our next question is, what is the function of COL7A1?
Yeah, so it encodes a trimeric protein that's secreted and then assembled at the dermal epidermal junction. And so it forms an attachment that essentially anchors the outer epidermis to the underlying dermis. And so if that structure is compromised, any friction causes severe blistering. And so it becomes a architectural compromisation of the integrity of the attachment of the skin. And so there are attenuated phenotypes or severe phenotypes, but it becomes something where any friction, as minor as friction from clothing, can cause blistering because of the loss of the architectural and structural integrity afforded by collagen 7.
Great. And then we have one last question in the chat. Do you feel that fetal insertion will be best for many of these rare diseases? And what might make this feasible?
Yeah, I think that's a great consideration. So the slide I shared with the lysosomal storage disease, there's one of the pioneers in lysosomal storage disease therapy painted a good picture where he said that if you think of lysosomal storage disease, a bathtub filling up with water, and the disease mutation is the plug. And as the water fills up, if you pull that plug, the water will drain, and that timing becomes important, because if you can't pull the plug, the water starts to overflow, causes flooding. Now you've flooded the floor. Let's say the bathtub now falls through the floor onto the stove, starts a fire that then starts the neighborhood on fire. At that point, if you pull the plug, you have this secondary cascade of disease pathogenesis, and pulling the plug or providing the transgene in that context may not fully resolve. And so I think that it really is guided by the accessibility of the target organ or organs, and then what are the secondary pathogenic considerations that are related to time. That a number of these genetic conditions really trigger these very severe secondary events that a primary gene correction may not fully address those. And so I think that early intervention may be desirable in that context, but I think then that will prompt certainly ethical and other deliverability considerations for, in that state, what are the true potential occurrences for off-target editing. And if you do that in early work with CRISPR showed that if you edited in the developing mouse embryo, the off-target effects could be magnified. And so I think that these things lay with one another, but I think that the secondary pathogenic drivers may not always be directly addressable. I do think that the exciting aspect of the multiplex capability of CRISPR may offer solutions or potential strategies for that. And so I think that the disease, the accessibility of the target organ, and the intervention will all coincide to guide what the best practices are and when to intervene.
Fantastic. Thank you again so much for speaking with us today and for answering the questions. We all learned a lot, and it's great to see you.
Thank you all so much.